Hello, everyone. We're so happy to have you here today. Welcome to our Ask the Expert series. If you're new to our webinar series, welcome and thank you for joining us today. Criterion Edge runs multiple informative webinars throughout the year. For today's live Q&A topic, we'll be discussing clinical data for IVD. What is it and how do I find it? Our first presenter today is Laurie Mitchell, founder and president of Criterion Edge, a global medical and regulatory writing firm serving the medical device, pharmaceutical, and biotech industries. Laurie has over 20 years of experience in medical writing, safety and pharmacovigilance management, and regulatory reporting. Having provided regulatory and medical writing solutions to many pharma and medical device companies, both large and small, she is a proven leader in designing practical strategies to meet current global regulatory challenges. Laurie is a published author and holds a Master of Nursing from UCLA. And also joining us today is Dr. Sarah Chavez, Principal Medical Writer with Criterion Edge. Sarah has a BS in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. And now I'll welcome Sarah to give an overview of our session today. Hi, Sarah.
Hi. So in today's session, we're hoping to answer some of your questions regarding aspects of clinical data. So specifically, how to generate, collect, analyze, or assess clinical data pertaining to your device, how to make the process of data collection, extraction, and presentation more streamlined, just some of our helpful tips, and then also discussing how to effectively present that data and within the PER template.
All right, let's get to our first question. Sarah, let me know when you can see it on your screen.
Okay, yes.
All right. This audience member is asking for some basic concepts of clinical data. What can you say about this, Sarah?
So just to sort of introduce the topic for folks who are brand new to this area, clinical data is going to be data pertaining to the use of your device in a clinical setting. So that's going to be a new requirement for IVDR, mostly in that up until now, you've been expected to present analytical data, but not necessarily how it performs in actual real world clinical settings. So typically, the way we would approach this in an MDR performance or an MDR CER, you would be identifying all of the sources of available clinical data that you can collect. So that could be published data sets, that could be some of the different registries, and that's often going to rely on some of the in-house data that you've also collected. So you are going to want to be aware of the multiple different sources of your data and ultimately how to present it within your report as a whole, how to make sure that you're being clear about the source of each of these different data sets. But just generally speaking, they kind of fall into the classification of being either in-house and sort of publicly available versus the in-house data versus the publicly available data sets.
All right, great. Let's get to our next question. This person is asking, what are the options for gathering clinical data of a device without the need of doing a clinical study?
So yeah, this is where those two different data sources really come into play. If you're looking for published data sets versus your in-house data sets. In this case, your options for gathering the data without the need of doing a clinical study really depend on whether or not anyone else has done those studies for you. So if there hasn't been any data published on your device in a clinical setting, that will necessitate some source of clinical data. So really you're relying on anything that you can identify in published literature through systematic literature review as going to be your primary data set.
All right.
Olivia, hi Sarah and Olivia. This is Laurie, everyone. I wanted to just jump on really quickly to this one and say that one other option is, so your manufacturer held data that Sarah is referring to is your best source of data, clinical data. You know, and I think we saw even a couple more questions about this. We may discuss it further in this Q&A session. But manufacturer held data is your most dependable source of data. You control it essentially, with the exception of your PMS data because it comes in passively. But think about what IVDR is now asking you to do, which is a post-market clinical performance follow-up studies. So that's again a source of data that you can collect proactively going forward that speaks directly to your data, your safety and performance objectives. So you're in control of the collection of that data. That's going to be a valuable source of data going forward for you. So be sure that you're laying down plans for your PMPF studies going forward right now, based on your safety and performance objectives that you're identifying for your device. So I wanted to raise your consciousness about that and other sources of clinical data that Sarah's already mentioned. But manufacturer held data is your most valuable data and usually your highest level of evidence data. So I just wanted to jump in with a couple of comments on that. Thank you.
Yeah, thanks, Laurie. Here's our next question. How do you address off-label clinical data for your IVD device?
Well, I can speak to this just using some examples actually that we've seen in how this is handled in the MDR world. Typically in a CER, if we are identifying data sets from off-label use, we would often exclude that as part of our screening process for the particular device. In the event that your device is, let's say there's very, very limited data, you may want to include it, but with the caveat that these are off-label uses. So you would want to make sure that you are distinguishing whether this is a current on-label or off-label use of that IVD. So I would I would represent that data as it is, that it is an off-label use. But what that could also tell you down the road is that maybe you're seeing quite a bit of data for this one particular off-label use that may inform your future changes to your IFU or the need to conduct additional in-house experiments to validate that off-label use.
Great, thanks, Sarah. This audience member is asking, for the IVDR, is it possible to use clinical or performance data from a similar device to support the performance evaluation?
So I think for this, it really depends on how you're defining similar. And this is something that we come into quite a bit as we're speaking with clients. From our point of view, similar could mean a competitor, but it could also mean an equivalent device. So you would definitely not want to use clinical performance data for a competitor to support the validity of your own device. You know, the competitor device data will be in the SVR in the one section where you're going to establish the competitive landscape. If you suppose you don't have any data on your device, but you have been able to successfully establish equivalence, in that case, you could use clinical data from the equivalent device to present within your CPR. But you must also make sure that you've truly established equivalence for those. If it's just a similar technology, but you're unable to establish equivalence, that's going to be a gap, and that's going to get picked up on pretty quickly.
Yeah, this is Laurie. I'll just jump in there saying, Sarah, that answer was perfect. And I'll summarize one last time for emphasis, is that in the evaluation, the performance evaluation report of the subject device, the goal of that PER is, as I always say in our webinars and anyone who will listen, the PER ultimately has one job to do, and that is to, at the end of it all, it will establish the safety and performance of the device under evaluation, the subject device, as it relates to IVDR. You're driving towards answering that one question. Is your device safe and performs as intended in the clinical setting? So with regards to that, then the similar devices are your competitors. They're, as Sarah said, they're going to be used to help accept the acceptance criteria in the competitive landscape. So if a similar means a competitor, as Sarah said, that goes to speak to those data, help to establish the acceptance criteria in your SVR. If you're talking about an equivalent device, an equivalent to the subject device, then there is a whole section of the PER where equivalence must be established. And then if you successfully establish equivalence between, say, the two devices, then, as I always say, data for one is applicable to data for all. So that's why we thought that this was a great question to include, because it drives home the on-the-ground differences between a competitor device, even if it's similar, and an equivalent device, which is equivalent to your subject device. Thanks, Olivia.
Yeah, thanks, Laurie. Get to our next question here. This person is looking for some insight or suggestions on the clinical validation sample size. What can you say about this, Sarah?
Honestly, this can vary quite a bit. And in many cases, it just depends on your particular device. If this is, and basically how commonly it's being used in a clinical setting. So if it's the type of device where the average study is only going to have a dozen patients because it's rarely used in clinical scenarios, you may set your limits much lower for what you would include in any given experiment as far as your inclusion and exclusion. For some tests, you may want to include things like case studies, where even it's a sample size of one, if it informs on a particular risk or a cross-reactivity or an interference or something that's been newly identified regarding your test. So it really will vary from device to device. I tend to extract all data sets initially and make note of the sample sizes. And then if you see a clear trend towards these larger data sets, those are going to definitely be appraised at a higher level compared to something like a case report. But the sample size itself, there isn't like sort of a set rule as to how many samples need to be used in a given study.
Okay, here's our next question. This audience member is asking how to determine criteria of data extraction.
Well, actually, we went through this in quite a bit of detail in our webinar last week. So definitely check that out if you're struggling with this issue. Just generally, the overall takeaway from the webinar that we presented was that you're going to collect data on many, many sources. So you're going to have a lot of different clinical data, and you're going to present that information separately. You'll then establish a range. So of all the articles identified, the range was between 90 and 100% for sensitivity. The acceptance criteria will then be derived from either the upper level bound or the lower bound of that range, depending on if you're talking about a safety or performance criteria. So, but yes, we went through this in quite a bit of detail in our previous webinar. So feel free to check that out if you're still a little bit unclear on how exactly those ranges are determined.
So I'll jump in here to tell the audience that this is a very common question that we get from our audience members, and that incoming questions into Criterion Edge is from your industry, because you're newer to this whole concept of acceptance criteria, whereas in the medical device world under MDR, that has been an evolving conversation over the last few years, especially probably the last 2.5. And so it's new and burgeoning area of focus in our IVD clients as well. It's like, what are acceptance criteria? What does it mean? How are acceptance criteria different than safety and performance objectives? And where do we get acceptance criteria? And what does it mean when we have them?
Mm-hmm.
And so the short answer to just build on what Sarah said was acceptance criteria are derived from competitor literature, not your own subject device. They are derived and then they set ranges of the criteria as it relates to your safety and performance objectives. Sarah mentioned sensitivity. So say that that is one of your safety or performance objectives, the sensitivity of your device. So, and we've established that your competitors are these 5 competitors that are most similar to your device. In other words, this is sort of the state-of-the-art landscape. So we would do a systematic literature review on those competitors that you've identified for your device under evaluation. We would do a a competitor device search, screen, and then selection of appropriate articles that have data in them on your same safety and performance objectives, but for the competitors. So we're going to be pulling sensitivity or performance objectives or whatever they would be, and their data. So maybe, as Sarah said, the range of sensitivity might be between 90 and 100%. That's in the competitors. So in this table that's created that documents all this search and the information that's derived from all the competitor literature, the end result may be that of the 25 articles that were identified for as competitor device searches, articles, the data in that for sensitivity the results ranged anywhere from 90 to 100%. So that's an acceptance criteria range. And there would be safety and there would be performance criteria as well, all expressed as a range within the competitor device. Now, you use those bars as a way now to compare your own subject device data to the state-of-the-art landscape, but you can't use a range. What we have done in the MDR world is that if it's a safety objective and you have a range of 90 to 100%, your device must be at least as good as the top range of your competitors. So if it's, let's just say if it's 90 to 95%, you must come in at least as good as 95 or above for safety, or below, below the top range in safety. If it's a performance objective, your device needs to be at least as good as the bottom of the range from your competitors. So I don't know if that confused people, but that's you pick a lane for your safety and your performance. And safety is your device must be better, at least as good as the top range. And for performance, it must be better than the bottom of the range for your competitors. I hope that helped. Send in your questions if you need more information on that, or watch the webinar that we did last week.
I actually created a whole table and sort of a mock data set just to help to better illustrate this, because we get a lot of questions on this.
Thank you, Sarah, for letting me jump in.
All right, our next question is kind of a big one. Sarah, how do you effectively present your data in your performance evaluation report?
I think first and foremost, if you're talking about it very high level, you want to make sure we use a tabular format. It's going to be very easy for the notified body to review, to be able to quickly glance at it, to make sure that you've got everything very well organized in a table. It'll make it much easier to compare between each of the different articles that you're including. But also remember that you've got different sources of data and they're presented in different places throughout the PER. You're going to have a section for your competitor data in that competitive landscape in one part of the PER, and then you're going to have your clinical data in another part of the PER. And you're even going to want to distinguish between your manufacturer held data and the published data sets. You're going to want to present them in different sections. So just high level, that tabular format with a text or some sort of a narrative to summarize what the table is actually showing you, but also keep in mind where you're putting that. That is just as important to maintain that order and the ease of readability for your notified body. You don't want them to have to dig through data sets or have them sort of jumbled up or somehow misorganized.
A couple of other hints that I would give. Thank you, Sarah. Especially that last emphasis on clear presentation and clear telling of the story. So I know the questioner may be thinking, well, how should I present the data in the performance evaluation report? And Sarah talked about a tabular format is the most effective to actually present the actual data. But in the analysis of the data, that's what they're reading. And so remember to be clear, concise, state your conclusions clearly, and even sometimes repeat them. Sometimes we tell, one of our tricks is that we tell the notified body reviewer 3 times what we just found out. It's like, yes, these are equivalent, or yes, you have met this bar. And yes, again, we are restating something is equivalent or it's met a certain bar. We really sometimes unusually call out a conclusion. And that is an effective presentation of your data and conclusion as well, is state it overtly. These reviewers are very tired and hungry and cranky, possibly. And they're trying to move through an enormous document with as much speed as possible. So one effective way to present something is state it clearly and obviously up front and repeat it and make it easy for them to come to their own conclusion as well, like through tabular formats. So that's my 2 cents on that.
Yeah, well said. Thanks, Laurie. Here's our next question. If the connection between the analyte and the condition is established, how much research and clinical data is then needed?
Well, actually, this comes down to sort of the purpose of that connection between the analyte and the condition. That is part of your SVR. So that's going to be sort of that initial justification that that analyte can, in fact, detect and validate that condition. So you're going to use clinical data regarding the ability of that analyte compared to other methods for doing that test. That's part of your state-of-the-art section within the SVR. But you're still expected to collect your clinical data, establishing that analyte, the association between the analyte and the condition is sort of step 1A. Once that's been done, you're still expected to conduct the reviews to identify any sources of clinical data to support the use in a clinical setting. And I think that comes down to sort of some of the inherent differences between IVDD and IVDR. Typically, you would sort of stop at showing that analyte association. That would be sort of the main purpose in IVDD. Whereas now it's that focus on that clinical condition, the clinical relevance. How does it's great if this performs well in your laboratory under very controlled settings, but how does it perform in the real world compared to your closest competitors? So really there's sort of 2 separate steps within the PER process. But in all cases, even if it's a well-established association, in that case, it might not be that tricky to say that, that you can detect HCG and that detects pregnancy. That association is very well established. So you would still explain sort of the process, why that is a good analyte, how it's detected, how the test works, but you would still need clinical data regarding how that test actually works in patients.
All right. This audience member is asking, what are the acceptable and unacceptable sources of clinical data? What can you say about this, Sarah?
I think first and foremost, you want to make sure you can document where you've identified the data sets. We want to collect high quality data. So this kind of comes down to the appraisal levels. We would use systematic literature review to identify sources of clinical data that have been published and sort of meet those higher levels of evidence. We would steer away from any claims that have been put out. So a claim is not the same as data. Your marketing claim may state our test is the fastest on the market or gets results, 10 times faster than the other. If you don't have that data in a published data set, that would not be an acceptable source of clinical data. That's a marketing claim. So, and the same with your competitors. You would want to steer away from claims that are not supported by a publication. But in some cases, you may be very, very limited by sources of clinical data. So you could still use conference abstracts or posters, for instance, in those scenarios, but you would show in your appraisals that those are actually lower levels of evidence compared to something like a randomized controlled trial in a peer-reviewed article. So we tend to steer away from any reviews that provide sort of aggregate data presentation that have not been a systematic literature review, because the review itself could be biased. But again, you could, as long as that is properly appraised and it's clear that there is a limitation to that particular data set, you could still use it. I think acceptable sort of is defined by how transparent you are about the source of the data and the level of evidence of that data.
Nice. Very well said, Sarah. Here's our next question. This audience member would like some advice for how to meet the ongoing clinical data requirements for new EUMDR clinical benefits, including safety and performance claims.
I think the answer for this, even though this is framed in terms of MDR, I think the answer is pretty much the same for IVDR as MDR. Really, it comes down to having that plan in place for post-market surveillance and collecting, having an ongoing data collection, but also using that to potentially identify gaps in your own data sets where a new study may be necessary. So this will really tie back to the purpose behind your PMS and your PMPF plans in the IVDR world, just to ensure that if there is a new clinical benefit that's been identified or a new safety and performance claim that's being routinely published, you would want to ensure that you are reflecting that as part of your ongoing efforts to collect new data. If everyone else is identified a safety and performance claim or a safety claim, you should be also adjusting your planning accordingly to start to also identify data relevant to that safety claim. So this can come up in different drafts of like from one CER to the next, where safety and performance claims may change a bit from version to version based on these sort of ongoing or new data that have been identified.
All right, great. We have just about 10 minutes or so left, so if you're still holding on to your questions, go ahead and send them in using the Q&A button, and we'll try to get to as many as we can. Here's our next one, Sarah. How is generating, collecting, and analyzing clinical data for IVDR submissions different than for IVDD submissions?
So really this, in this case, IVDR is going to have much more of a reliance on that clinical data. Up until now, you've really been focused on collecting the performance data in terms of the analytical performance. So the main differences, I think, in terms of what we're all familiar with in IVDD, the analytical performance is kind of where we've stopped. You establish the association, you provide information regarding your purpose and things like that. But that clinical data is a brand new requirement. So we have, and particularly how it relates to risk. So that's going to be sort of the primary difference that I would propose, is that it's going to come down to how are we collecting data for real world scenarios, real world usage of our device in clinical settings.
Great. All right, here's our next question. Which are the most relevant clinical data?
So this, again, it will definitely depend on your device and the intended purpose of your device. So getting clarity on your intended purpose very early on is going to definitely be helpful. And again, we referred to this in one of our previous webinars several weeks ago, but the intended purpose statement is really going to tell you whether or not your device is being used to diagnose a clinical condition, being used to monitor treatment or response to treatment as sort of like a biomarker for prognosis. Those are all going to potentially have different types of data or different representations of data. So if it's just there to show that the ability to detect an analyte, to diagnose a medical condition, you're probably going to be looking at sensitivity or specificity or positive predictive value, negative predictive value. Sort of those are the standard ones. But if you're talking about things like prognostic value or response to therapy, you may be looking for data that are things like area under the curve or Kaplan-Meier survival curves. You know, there are good hazard ratios. There are other different ways the data will be represented. So if you know that your intended purpose encompasses both, you may have a particular test that can be used not only for diagnosis of a disease, but also for monitoring treatment or response to treatment. In that case, you may want to collect data on both types of clinical data or both sets of clinical data and be able to explain sort of like what the relevance is with regards to each of the different intended purposes.
Nice. Let's get to our next question. Here it is. This person is asking how to use surveys as PMCF.
Oh, Laurie, do you have much experience in this area, or can you speak to this?
Sure.
I'm not as familiar with how the surveys fall in.
So in our, and this again says PMCF, so that's the MDR world. So we will translate that to PMPF for our IVD audience today. Surveys are just what they say, and we've worked with some of our clients to build surveys that then they can send to end users, for example, or their key opinion leaders, or some of their key investigators or physicians that use their devices. It is a low level of evidence, that is for sure, because it's basically asking for unvalidated data from the point of view of an end user. So it's a lower level of evidence, but you can certainly design a PMPF survey, put it into your plan, your PMPF plan, and then collect data on a routine and ongoing basis, which is proactive. And that survey should speak to directly trying to collect data from those end users or whoever you're sending the survey to, collecting data from them that are specific to your safety and performance objectives. The key here is that your safety and performance objectives that you identify for your device or analyte, everything that you gather as far as data should be in service of collecting data on those safety and performance objectives, first and foremost. Obviously, you're going to want to do rigorous studies all the way through surveys like what this example is showing. But either way, you should always be asking the right questions, whether it's an endpoint in a clinical trial or a question on a survey. And those questions need to relate back to ultimately providing data on your safety and performance objectives. Those are your data. So that's my thoughts on that.
Great, thanks, Laurie. All right, we have a few minutes left, so we'll keep going with the questions. Here's our next one, Sarah. How would you tailor the PER to your data, and should you keep a standardized template?
So there's going to be a standardized template regardless for the PER. There are certain key requirements that should be included regardless of your type of device. So across all classes of device, they're going to expect to see certain elements within the PER. Now, the level of detail is really when where they're going to diverge primarily. You know, if you have you're going to make sure that the presentation of the data is proportionate to whatever risk class you fall within. So clearly being a class D or something, you're going to be held to more rigorous standards in terms of presenting your quality management system, your post-market surveillance, you know, your general safety and performance requirements are going to be more rigorous. But you're still expected to have the same types of information contained throughout. So you would still have your your SVR, APR, CPR, you would still have those sections. It's just the level of rigor that you will need to justify risk, really, and to justify your choice of safety and performance criteria may vary depending on how many indications your device has, how many purposes, how many ways or patient populations that it's used in. It sort of will vary depending on the level of sort of utility. How many scenarios can you use your device in? So that's where having that template and an organized structure will really, really benefit you. But overall, the template will be similar in terms of like the flow of the overall document.
Great answer. All right, this next audience member is asking, for an intended population and clinical trials, how do I bridge data from existing clinical trials?
So this kind of shows the overall purpose of that post-market surveillance and really why this ongoing monitoring, what its intention is. Because if you have a test that really was designed to meet a specific need in a specific intended population. Let's say it was used originally in adults. Now you're starting to see more and more data from your competitors also using the competitor devices in pediatric populations. That's a distinct population. If you only have internal manufacturer held data on adult populations, you would want to then plan for current or future trials to be focused in on that pediatric population. So, and even if it's like, let's say it's for testing for a new medical condition or a new purpose, all of those things are going to sort of draw information from your review of current guidelines and recommendations and your review of what you're seeing within that competitive landscape for your closest competitors. So it's meant to show that sort of that literally that bridge, how you can collect that data from what you're identifying in your current search and use it to show improvement, and that you are putting in the due diligence to identify this type of data moving forward.
All right, Sarah, we have just a few more questions. Here's our next one. This person is asking, what information should be included in the PER for an analyzer, considering that performance data is related to the reagent?
Yes, so this can definitely be challenging. In our experience, the way that we've been handling is that handling this for something like an analyzer or a component of a system, we would include information about the reagents as well as the systems that are being used for the different competitors. So that way you can make that sort of comparison directly. You can show that while the article may not report on data for that analyzer specifically, you can compare if two different analyzers are used with the same reagent for these two different studies. You can show that sort of association. So it may be more challenging if it's something like a software or a hardware device that doesn't necessarily include direct data, but that indirect data, as long as you can provide a justification for why and how you're using it, that's going to be the best approach as far as like still establishing what types of performance data you can collect.
Great, thanks, Sarah. Here's our last question for the day. How do I justify that I need no clinical performance study in cases that don't need it? In what case would the data be necessary?
From our understanding, the Class A devices, it's generally acknowledged that the clinical performance report, the CPR, is not going to be necessary for a Class A. So in place in that section where that would have lived within the CPR, you can use some templated language or some sort of a general explanation as to why your device why a clinical performance study for your device would not be required or necessary. But in the majority of cases, it's expected for Class B and above that you would have clinical performance studies and clinical performance data within that CPR.
So Sarah, I just wanted to clarify that with what you said is that if the device is a Class A, that no CPR is required. Is that what you saying?
Yes, the majority of
or a PER. What? Say that again.
Oh, yes. So for a Class A device, you would still be expected to have all of the requirements for the PER, but in place of a formal clinical performance report where you're going to be looking for very detailed data, you would be you would include some sort of a language or an explanation as to why your device is a Class A, why that clinical performance study isn't necessary. So if it's a general reagent or something along those lines, you can just state that these are used under a wide range of circumstances and a wide range of clinical scenarios, and data is rarely reported with regards to that specific IVD. But you would still be expected to present your analytical performance data that you would have through your in-house studies. So that's where you would still collect data on your competitors, anything that's been published in the literature with regards to clinical or analytical performance, but you would not necessarily be expected to have that same level of detail as you would for like a Class B or above.
All right, thanks, Sarah. I think that's a great place to end on. That's all the time that we have today. If we didn't get to your question, we will go ahead and reach out to you by email, but I hope we were able to answer most of your questions today. Thank you again, Sarah and Laurie and everyone for joining us. There were so many great questions answered today. Take care, and I hope to see you at our next webinar.